A lot of research in genomics has been performed in largely European descent cohorts. But we know that the overall human population is extremely diverse. And we really need to understand the role of that diversity in biomedical research and how these differences might influence differences clinically and how diseases may manifest, whether they manifest more aggressively, differences in the way individuals may need to be treated, taking a more personalized approach. Los Angeles happens to be the second largest city in the United States, and Los Angeles County happens to be one of the most diverse populations and communities in the United States. So USC and the Keck School of Medicine is uniquely positioned geographically, right, to take advantage of a vast array of genetic diversity and understanding how differences in this genetic diversity might be impacting other differences that we see in health.
USC Translational Genomics is truly special. We've been focused on bringing in individuals from all types of backgrounds, cancer biology, neurobiology, informatics, and bringing them together in this kind of area where we can collaborate, work together, and make these advances hand in hand with the physicians that are part of this great medical school.
The technology is really the thread that connects all of us together. That's why we're all here. We want access to this. We want to be able to be the super users of these technologies and collaborate with other people, other clinical departments, so that they can work on this too.
Now to be able to use technologies that allow us to sequence the genome in single cells, right, and understand the differences of these different cell types is one of the true advancements in genome science. And taking that a step farther, being able to do that on an actual tissue section to understand not only the various components of each cell, but how the cells are communicating with each other in space.
What we're doing with the Bench with Bedside initiative, which is a play on bench to bedside, is that we are trying to incorporate patients and engage patients more in the translational research process. We want to inform them of the results of the research as we conduct the research so that they're aware and that they know what the sample that they gave us has done for us and for the research endeavor. What we're doing with Bench with Bedside initiative is that we're actually ensuring that we represent the diversity that exists within our own catchment areas.
I'm working in cancer genomics and cancer health disparities, and basically what we are trying to do is to identify different mechanisms the cell wants to adapt in order to retrieve the health status. For example, if we have African Americans and Latinos with the same disease, sometimes signs different genomic characteristics, the prognosis of the disease is different, even if it is the same disease. And this is something that we want to understand. What is happening inside the DNA? And not just inside the DNA, but also in the microenvironment of the cell that make people with different ethnicity be more susceptible for higher grade tumors than other ethnicity. And this will really help us to better decide which treatment or which medication will really hit the tumor in the best way. So that is precision medicine.
I'm actually going to be moving a lot of these technologies into the neurological disease space. So I work on Parkinson's disease, Alzheimer's disease, in addition to psychiatric disorders like schizophrenia, depression, bipolar disorder. The single cell technology actually allows us to look at individual cells separate from one another. So rather than getting this pool of all of these transcripts, we can say, oh, these genes were expressed in these specific cell types. This becomes very important for research in the brain because there are a lot of cell types in the brain, specifically a lot of different types of subsets of neurons. Being able to look at these individual cell types separate from everything else allows us to ask very specific questions we weren't able to ask before. With the spatial transcriptomics added into that, now we can not only look at individual kind of cell populations, but we can see how they're communicating with one another.
We tend to look at diversity not only from the standpoint of our interest in diversity in biomedical research, but also from the standpoint of workforce development. And we're very intentional about making sure that opportunities are available for all people.
A very important thing that we do is really students' personality, their experience, their motivation. And we recognize it, we value it, and we take advantage of that so that we can build a team. We are diverse, we have different way of thinking, we have different idea about how to do things, but we work together. And I have never been in such a wonderful, nurturing, supportive environment.